So what's really cool about research is that all research kind of builds on each other. So different concepts and different techniques. One of the strengths of being in Hao Chang's lab and being in an environment like Stanford is that there is a lot of crosstalk and there is a lot of diversity in the research programs. So a method was developed in the Chang lab before I arrived called ChirpMS. What ChirpMS was doing was basically, it was an RNA-directed proteomics approach. And essentially what that means is that if you have an RNA of interest, what you would do is pull it down and see all the different proteins that are binding with it. This is a technique that was initially used to assess Xist. At the time, the field was very interested in seeing, and is still interested in seeing, how Xist functions. In this protein set, there are many, several dozen proteins, but about a third of them looked very familiar from the autoimmune angle, because actually a third of them were known autoantigens with connections to autoimmunity. That's really where the initial sort of seed was planted, like, oh, okay, if this complex being in every single female cell, every single XX cell, can somehow contribute and help explain why females develop autoimmunity so much more than males do. Again, it's a hypothesis that it was a little bit risky because the answer could have been yes or no. And so to see that the answers seem to be trending towards, yes, there's something there, that was very exciting.